זה מישהו בחוץ? כן. 12 וחצי שנה? 12. 12 לא גרמני. זה השיעורים בעברית או באנגלית? אנגלית? שאני אגיד להם שאתה מתחיל? כן. כן, אפשר. פיזיולוגיה, קצת. הזמנתם מה? סטודנט יודע לשים שמות על פרוטוקול? לא, אבל אני הבנתי שזה העלה להם בבחירות ממש יופי. כאילו יש לעשה רושם של זר מסתבר, מסתבר שאנשים, אולי יותר ילדים, אבל אנשים מתים על זה. לקנות פחית עם השם שלהם. שם של הסדנה.
שלום, אני הלכתי לבילור, תחתום לי בבקשה על היד פה, כוכב קולנוע, דוקטור אמיר עמדי, פרופסור, סליחה. סליחה.
לא תגיד שלום.
אולי הוא קצת כזה.
כן, בסדר. גמרנו את הכנס, הלכתי, הלכתי פה 200 סרטים מכל העולם, שבוע היה וורקשופ, 30 מרצים קוגניטיביות, די
צילמת, תמונות, יש פרסום, אתה עושה דברים לבד, אתה חושב שאתה חי שם וזהו. אתה לא חי שם, זה בסדר, זה שאתה שומר על זה שאתה אימריק ומדיקל סקול ואלסק זה אחלה. אבל בוא תתן לנו גם...
לא, אני מתאר לכם שנייה, אני אשתתף.
לא, בשבילך, מה אכפת לך?
אין לי כוח.
תראה, תכניס את זה לאיזה...
אני כבר, זה מה שקורה מעצמו קורה, זה מה שלא...
בוא אני אגיד לך, תכניס את זה למין איזה מתכונת קבועה. יש לך 3 הורים. בסדר, הם תמיד קיימים. וכל אחד תקבל ממה שאתה יכול לקבל ממנו. אז בא לך, אתה לא... אתה מבין, אני לא...
יהיה הרבה יותר קל כשיהיה...
אני לא אומר שאנחנו יותר טובים, או אתה שייך אלינו יותר ממה שאתה שייך אליהם, או... ממש לא.
לא, זה לא מה שייך. את המעט זמן שיש לי בעבודה, אני רוצה להשקיע במעבדה, ולא איזה עבודה,
לא עכשיו קצת בארץ, רגוע קצת?
כן, יש לי קיץ מאוד רגוע.
כמה משפחה בטח?
כן,
יש לך
ניקח חופש של שבוע.
יש לך אומר שהגירושים זה יהיה עוד יקר. לא יודע מה זה.
בסדר, בינתיים זה תחת שליטה, אבל לא, כיף את הקיץ הזה. טוב, בזה רפי, יאללה, שיהיה יום טוב. טוב לראות אותך. Okay, so we have... Could you move the microphone by any chance? Is it here? I think it's going to be fine.
close it. Or is that PowerPoint?
I think with such a small class it's going to be fine. Can you hear me?
No, but the problem is the video.
Okay.
There are people sitting at home now and watching you.
Yeah. That's stressing.
Thousands of people.
So that's not good. I put a lot of unpublished data.
This is something you won't, not clearly visible on the TV.
What,
Yes,
medium?
You know, you can't see the presentation, you can't hear it.
Now I feel it's more official, so I'm going to be more serious. Okay, so my name is Amir Amedi. I'm part of Elsc and the medical school, so my lab sits in the medical school at the moment. And since I think we will talk to mainly, I think, if we have, it's like 45 minutes and then a break, and then, or we take it straight. Okay, excellent, a break. So what I think I'm mainly going to talk about is brain plasticity and stability in the brain. And we'll talk mainly about basic research, but connected to visual rehabilitation. Now, since I think you get very little, since this is what I work on the human brain, we'll talk mainly about plasticity in the human brain. but also I think you don't hear enough about human studies. But I will try also to connect it with a couple of examples from animal studies and try to compare and see generalization. I will show you some things which are textbooks and some things that are really controversial and new in terms of our view of brain organization and plasticity in the large scale. I'm going to talk about large scale organization using neuroimaging technique, TMS, etc. But I will try to connect it also to animal research and try to see if it holds water. But when I will get to more controversial parts, I'll mention it, that this is what is textbooks and what is not. So let's start with, and I'm going to present a lot of data from the team in my lab. There are, by the way, I'm going to present quite some data from Uri Hertz, Noah Scharia, and Nadine. 3 of them are from ICNC Elsc students, and the rest are coming from various backgrounds, computer vision, neuroscience, psychology, so it's a very interdisciplinary work, and I'll try to give credit to who it is. So let's start with a very, very basic one definition of cortical plasticity. And when we talk about, and of course people talk about cortical plasticity from the level of molecules and changes in the genetic transcription all the way through large-scale changes in global maps and global organization of the senses in the human brain. So we would focus today on this large-scale organization. So we talk about changes that occur in the function and the organization of the cerebral cortex as a consequence of experience. Okay? And I think one of the most influential work in this concept of plasticity and also critical periods for plasticity is the work by Jubel and Wiesel, in which they basically patched one eye, had animals have one eye closed, so without receiving any visual experience, and one eye open, and during different periods injected tracers and mapped basically the ocular dominance columns in the primary visual cortex. Did you learn about it already? So you know this works, so this is just a quick reminder. So this is the normal development of the ocular dominance. So in black stripes you get input coming from one eye, and in white from the other eye. One is labeled and one is not. And if you are closing the eyes for a critical period, even if you have a lot of visual experience after that, this is irreversible. You get neurons that respond to one eye take over almost the entire primary visual cortex. And even if you have tons of visual experience, the eye is blind. This eye that was closed for this critical period is blind, and you can never recover. You cannot have plasticity. So this is the concept of critical periods. And if you are doing it, closing the eye in a later age and doing the same thing, you do get recovery, or in principle you can get it. Another exactly. So the neurons that usually respond to one eye, so they are dominant. Let's say the right eye. If you don't get input from the right eye because it's blind during the critical period, the neurons would change their dominance and would now respond to the left eye. No, so this is not part of their work. Their work was during this critical period. So it's not taking over. And there is well, they didn't do this experiment. Okay, so we will talk about about adult brain plasticity. Okay, another work which was looking at what happened without visual experience, but this time with complete blindness. This is one of the key studies of this. This is in a brain of a cat, and what you see here is that, like in the human brain, the cat brain have a visual cortex, somatosensory cortex, and auditory cortex, but there is also projections into the prefrontal cortex around the anterior ectosylvian sulcus of maps of the three senses. So the three senses, and this is the common wisdom or the common knowledge of the large-scale organization of the brain, both in animals and in humans. This is still the textbooks of today. That you have unisensory areas, okay, and so the visual cortex, the occipital cortex gets information from vision, the temporal cortex gets information from sound, the parietal from touch, and so on and so forth. And then you have projections into higher order areas, and in these higher order areas you get still unisensory maps. This, at least in the case of the cat brain. This is work by Joseph Rauschecker. And then you start to get also some overlap and convergence between them in these areas. And then you have other areas that presumably are combining and binding this information and process this. Okay? And what Joseph Rauschecker showed, it's actually one of the first demonstration of this massive large-scale plasticity, is that if you raise the cat without visual experience, so these neurons that usually receive information from the visual cortex, there are two options. One, that they would be unresponsive to anything, okay? But what he showed is actually the auditory and the somatosensory neurons are presumably firing and then recruiting the other neurons that are vision by origin. And all these neurons that were originally visual are now auditory and somatosensory. Yeah.
What is the name of this region in the cat, and what is it? And does it have an analog in humans?
So the area is called the anterior ectosylvian sulcus. It's the area around the anterior ectosylvian sulcus, and it is not clear what is the analog in humans, but we are going to I'm going to show you a lot of data from humans from a very similar model. So we are going to get similar but different picture. Right, so this is a very good point. So what is cool about this research is that not only that Joseph Rauschecker showed that they are taken over by auditory input, but he showed, and I'm not going to show you all the data, he showed that these blind cats that were raised without visual experience have superior auditory localization, especially in the periphery, okay? And there is some correlation between the fact that there is more responsive neurons to these superior abilities, okay? So the fact that they have more cortex, it could have been also just that the neurons invade, and by Hebbian rules they fire, and then by accident they fire together, and then it increases the synaptic strength, and then they start to fire more and more, and slowly by having so much time without visual experience, these neurons become auditory or somatosensory, and it could have no functional relevance. So we will talk a lot about also functional relevance when we can. In this case, they showed that these cats not only have more auditory areas, but they also have better auditory localization. I'm going to show you more recent work that actually showed beautifully that these types, it's a different example. It's a deaf cat raised without auditory input, and looking on changes in visual changes, if they have superior visual abilities and this type of plasticity. So towards the end of this lesson, I guess we might get to this, or the second lesson. So this is a very good question, whether this changes not only these neurons fire for auditory input or visual input or tactile input, but really contribute to behavior. So in some cases it was shown and some not. Okay, I'm going to give one more question, and then I want to also manage to get to show you some data. Yes?
Can you just say what the abbreviation of the population is?
Well, it's the names of the different areas in the cat's anatomy, and I don't even familiar with it. Okay. So what I'm going to, so this is just a short, brief intro. I try to connect it to some of the stuff you might heard before. So what I'm going to talk about is about blindness and deafness as a model for studying brain plasticity and stability, just you saw here, but in the human brain. We can ask whether the blind brain learned in adulthood to process visual information conveyed indirectly via a different sense in a new manner, and how far can we push it from some of the program that we have in our lab. Then we will ask what happens in the visual cortex to this new sensory input that is functionally meaningful for the blind. So we were not only going to raise them with blindness, although I might show you some examples of blind visual responses to blind to processing in the blind without any special intervention. But in this case, we will try to teach them to see, to understand the environment better using a different sensory modality. I think it will be more clear when I show you the exact details. And we will ask what happened to this 30%, 25% of the brain, which it's a crazy number of neurons. What happens when they don't get the visual input from the eye and from the visual pathways? And based on this, I would try to suggest a new way to look on brain organization, which might be a little bit controversial, but we'll see if the data supports it or not. Okay? I will skip this. I'm not sure, given that you're interested in asking questions, I don't think we'll get into the pen-filled homunculi and the new ways to map the human body in the brain. That might have to wait for a different opportunity. Okay, so let's talk very, very briefly about vision. You already learned about the retina and the organization of the retina and the visual pathways. The point of this slide is to say that a lot of people treat the visual system a little bit like a camera, and there is a lot of similarities, indeed. So we have the image, we have the lenses that focus the image, the inverted image, we have the shutters and the iris, and so on and so forth, okay? But the point I want to make, by giving you one example, which I really like because it's an example that happened to me spontaneously, is this example. You use a lot, as a researcher, in the 10 years ago or 15 years ago, even more, but now still in other contexts, FedEx a lot. So I looked at this logo many times. You send manuscripts, you send materials, you get them back. And after hundreds of times I looked at this logo, suddenly I saw that there is an object there that I never saw before, okay? And it was like quite strange. What happened here? How come it be that it was there and I didn't see it, and now I see it? So how many of you can? Yes. So between the E and the X, there is an arrow. Okay. So how many of you actually see the arrow for the first time now? Okay. So it's usually what's happening in a crowd. About half or 2/3 of the people didn't see the arrow. But the thing is that now, what will happen is that you went through a really radical case of single trial cortical plasticity. Because now each time you would see this FedEx, you can't get rid of the arrow, okay? So next time you walk on the street, you see this truck. First of all, you see this arrow immediately. Second of all, you remember this weird lesson that Amir Ahmedi taught you, and that's it. And your brain changed forever, okay? So this is one type of how experience can change our brain. The second point is that nothing on your retina changes when you see the arrow, okay? So a lot of the processing in vision, it sounds trivial, but a lot of the is that we see with our brain not less than we see with our eyes. And then this gave rise to a field that it is called sensory substitution, and the pioneer was Paul Bacharita. And his idea was, yeah?
I'm sorry, but how do you know that there is no plasticity at the low level? Like, I don't know exactly the retina, but in gamma something is...
No, we don't know. The point is that in many cases, the visual, I can show you 2 hours of example that the visual input did not change.
Well,
I'm not.
Change,
Yeah. So the point is that if you look at the visual system in a naive, yeah, I'm just giving it as an extreme example, as a naive way, looking at like a camera. So the input is what matters. So this is just one example where the input can stay exactly the same, but our perception is very different. Now, since we have so many neurons and it happens so quickly and many times unconsciously, it looks like a very simple task. But the point is that it's not. We all the time build.
My question is about how do you know it's perceptual or not the sensory input? Like in this case, but never mind.
Yeah, of course. It can be that we perceive so we can do control experiments, etc. This is just a general example, but it happens a lot. And also, active sensing can be plasticity because the fact that you look differently and look for the information, we sample each image 3 times in a second, in a very quick saccade. If you look at the data that you receive, it's really very partial and very, very weird data, and we build the image around it all the time. This was mainly the point. And the point of Bach-Harita was, if so much of vision is central and require brain mechanisms, then why don't we bypass it and deliver the information using a different sense? So his idea mainly used tactile approaches. So there is glasses, and on the glasses there is a little camera. The camera captures instead of the eye. Most of the types of blindness are not cortical. Cortical blindness is something very, very rare. The reason is that we have so much of the cortex dedicated to vision that you need like massive bilateral strokes that usually would kill you. Okay, so there is some cases of cortical blindness, but most of the problems are in the retina and the eyes, the retina, and the pathways that leads the information to the cortex. So the idea was to all this blind individual, what we'll do, we'll have a camera looking at the image. Then he built a huge dental chair. So the technology was not with electrodes, with vibrators on the back. And of course, you know that 2-point discrimination in the back is about 4 centimeters. So you can't have a lot of pixels. But he managed to show that blind people can do object recognition and can do quite things. You have beautiful description of how, with extended use, they do what we call distal attribution. So in the beginning, when they would look at the vibration, let's say there is a bottle, they would feel dots on their back. And they would attribute the dot to tactile sensation on their back. But as the blind trained more and more, they stopped sensing the objects on their back. They start to attribute it to things that are in the outer world. Okay? So they start to have distal attribution to objects. So this is something really interesting, and they didn't look too much on the brain mechanisms of it. Now, yeah. So in this case, not, but we will now show, I will show you a lot of brain imaging of longitudinal studies of the brains before you start to. Oh, it's very simple, on the back.
So why is it as a visual?
Well, this is a good question. But this is, first of all, you can't say that this is visual, okay? And sometimes they would report, in late blind, they would report anecdotally that after extensive use, they start having visuals, like, it would start like flashing to them as a visual thing. But this is very controversial and debated. And some people don't. They just understand, and I would show you some examples from auditory sensory substitution. The point was not that Paul Bach-Harita didn't claim that he's giving them vision back, but they can understand things that, let's say there is something that I can't reach and I can't perceive, what is usually the case for blind people. They have the peripersonal space and they can sense the world around them using touch mostly, and only if things make sound, so they can understand things a little bit farther. So now they would look at it, receive the information by tactile stimulation on the back, and basically know that this is a bottle. Okay? So he gave some basic examples. Now, in my lab, what we do is we also use sensory substitution, but we use a little bit tactile sensory substitution, but mostly we use auditory sensory substitutions. And I know it sounds a little bit more complicated, so I would actually train you for 3-4 minutes on trying to understand visual input using sound. And the reason that we use so the idea is, again, that you have glasses, you have a camera. The camera is connected to some processing unit. This processing unit in the beginning was a computer or a netbook, and now it can be a dedicated processing unit or an iPhone or smartphone, etc. Then you have headphones that give the information. It could be regular headphones, but if the blind prefer, it can be bone conduction headphones, like in this case. So you would leave the ears open to receive information from the environment, and you would have the auditory signals vibrating on the bone, and this would transmit it to the inner ear, and through the regular transmission, and basically they would hear. And you can hear Mozart symphonies with this as clear as regular headphones. Okay, now the reason that we used, first of all, in general, sensory substitution are non-invasive. Now this is the point that you ask. Usually there is no visual qualia, so they can, and I would show you, you would experience now for yourself. You can, in the beginning, you understand what happens in the outer world, but you don't see. It's not that you have like a hallucination or dream in a dream or in a hallucination. You actually see the image. Okay, so it's not the case. Now, anecdotally, there are some late blind that use this a lot and start reporting that sometimes they would close the door and they would have a flash of the door in their brain. They would actually report something that is very close to visual qualia. But again, this is very debated and not validated scientifically in a good enough way. Okay? No. So this is, we can't even, exactly, this we can't even, we don't know, and I will show you a lot of data from congenitally blind individuals without visual experience, and we will ask how their brain organized in relation to vision, but we don't know even how to ask them. They say, okay, I understand what's going on there, but they don't have even a reference to vision. So, but in late blind, there is some, so for example, the woman that reports having like flashes of vision sometimes, she was sighted for 22 years, then she has some accident in a factory that she worked, and she's, in the time of the experiments, was blind for like 20 years. Okay, so, and she's the one that reports this. And by the way, there is a huge variance also in terms of visual imagery. Okay, we are not going to talk, with some studies on this, but we are not going to elaborate too much. But in terms of visual imagery, some people report, like me, when I try to imagine an object, let's say I'm looking at this object and try to mentally image it, I'm seeing only like really gray image on a black background, really, really poor mental imagery, visual mental imagery. And some people say, they look at this, close the eyes, say, I see it as a picture or as a TV. And there is some questionnaires that try to quantify this. So there is a lot of individual variability. It's also important to the question of perception and plasticity. So some of it might be in late blind attributed to this, but we didn't test it we didn't have a specific project to test this question of qualia. But it's something that starts to interest various labs that work on sensory substitution. Last question. And then two questions. And then we have 10 minutes of, I want to get to show you some data also.
The first one is how the bone conduction that we know the outer field is from 500 Hz to 3 kHz. How it interfere with the natural hearing?
Sure, that's a good question. And I will show you some movies that they managed to do quite complicated things while listening to speech. But this is another thing. This is a relatively new field, especially of the new auditory and high-resolution sensory substitution. So this is still an open question, how exactly they do this and how well can they do it. So you need to do control experiments of trying to perceive outer signals and inner signals. In a very unscientific way, you see that they manage to communicate, talk, walk, and hear things from the environment. And probably they do it by how we do it for different visual input. So if I have now, I have a lot of visual input here, and what I can do is shift my attention very, very quickly in milliseconds, okay? So probably what they do is they shift their attention when they do this, and then somebody talks, they listen to it, they bring it back. It's happening quite fast. So it seems as though there is no strong interference. But for sure, in some load, there is interference, and we didn't test it, okay? This is the second or the third already? Okay, this is the second. Okay.
I don't know what the processing unit you have made exactly, but how do you match between the recessive field of your input?
Okay, so we would, you have very good questions, but it's all coming, okay? So we actually would see the interesting question, or one of the most interesting questions, one very interesting question, is the transformation algorithms. How do you keep visual information in the sound, okay? So for example, the veteran SSDs, some of them we used, were all grayscale. So you convert the image into grayscale, and then you only transmit grayscale information. I would show you.
Again, it's very small windows, so how you convert very large window of visual input into very small.
So we will see it. We actually will train your brain to do this now, okay? If you're up for it. Okay, so the idea is exactly what you asked for, and of course there is many ways to do it. To keep visual information in touch. Touch is more natural, so it's topographic, and then let's say you have the letter. Letters is very simple to explain, okay? So let's say you have the letter V, so you have pattern on V on your back, or in the new sensory substitution that was based on Paul Bacharita inventions, they now have a tongue display. Maybe you saw it on YouTube or something. It's called the brain port, and it's like 200 electrodes that sit on the tongue, and the blind receive electrical stimulation on the tongue and can do quite cool things. Now, the key thing is that you need, in order to do it in the auditory modality, and very soon I would explain why the auditory modality, then you need some rules that keeps the topographic information in vision that is maintained in the retinotopic maps in the brain. So there is topography in the world that is maintained in the brain. So you need to keep this topography in the sounds. So you need some algorithms. And of course, this is one very interesting field. What is better algorithms? And very, very little research is done on this. We, for example, developed an algorithm that keeps not only the topography, but also the colors. I will show you in a second how. Very briefly, we don't get too much into the details. But there might be other more smart algorithms that keep more of the information or are more easy to learn. And this is a very, very interesting field in sensory substitution. I'm going to do some training on you, but we are not going to talk too much about the algorithms. This is a very, very interesting field, and not enough work is being done, and some of it is being done in the lab. But of course, there is still much more to do on this. The second would be to ask how the brain adapts to this information and start to represent vision again, okay? So this is what I hope to show you. So we are focusing on auditory sensory substitution. The reason is that, first of all, they are relatively cheap. So if you build this device and you have a smartphone, all you need is a camera that costs almost nothing today. Yeah? 50, 100, let's translate it to English, $20, $30, $50. Okay, you need headphones. If you want the bone conduction, it's $80, $100. If you want the regular ones, you have them already. And you just need a processing unit, and that's it. So this is really cheap, and it's very important because if you want later to try and use it for rehabilitation, there is a lot of blind in the Western world also, okay? We talk about 45 blind people that are fully blind in the world, 45 million, and then another over 200 million that are severe visually impaired, almost 300 million now. But most of them still are living in developing countries and are quite can't afford, for example, the brain port, which costs $15,000. $15,000, yeah. Okay. The second more important thing important for research, they are MRI compatible, so it's easy to give sounds in the scanner without and look at the brain how it responds to them. And the last point is that in contrast to tactile sensory substitutions and to site restoration, active site restoration efforts that we will, hopefully I'm going to talk a little bit about them towards the end of the lesson, like new ways to represent vision this through interaction with the neurons. In some diseases, there is still, the neurons are still intact. So for example, you can have diseases in which the photoreceptors are degenerated, but the neurons themselves are fine. Of course, you are blind because nothing is doing the transduction of the light information into electrical information. So you can either stimulate directly the neurons, or you can implant, for example, channels that respond to light. And there is a lot of effort in this direction now. But in all these cases, we are still talking about a relatively limited number of pixels. So for example, the most advanced, and I'm going to show you a little bit about this, advanced chronicle implant that is being used today for blind individuals is the Argus 2, and it has 60 electrodes. So 60 pixels, and in the retina. And here we talk about thousands of pixels. Okay? Yes. We will never get to the data. Yes. No, but this is great. But I don't mind. We can keep for another hour, several hours. But I think you have some lessons after that, no? Yeah. That's a very good question. And let's talk about it in the end, okay? But this is very speculative. But in principle, you can think about a situation. I like to give this example of, so just because you asked, to suggest the new James Bond movie, the next one, okay? It will call instead to your eyes only, to your ears only. And imagine that you have a camera that receives information that is across the bandwidth that we have in vision, let's say infrared. Or, yeah? So, for example, I can sense warm things around this, in the other side of this wall. I can walk in the corridor, have my vision, but also see in which place we have the bad guys, okay, and by the heat. And if my brain is trained on this, maybe I can see vision and at the same time have this heat map delivered to my ears and process both of them and increase vision. So this is a new field that is called sensory augmentation. We also play with this in the lab a little bit. There is already one company that is based on this concept, and a research, very beautiful research program. And the idea is that what they try to do is to deliver the north, the true north all the time. You have a belt that gives you tactile information about the north all the time. And you wear it. You wear it now for weeks or months. And the question is whether this would give you a natural sensation of where is the north and would improve your navigation and your orientation in the environment. Imagine that you have all the time the north, okay? Would you start to have better cognitive maps on the environment, etc.? And then look also on the brain plasticity and brain mechanism, very similar to what I would show you in our program of sensory substitution. So this is a very exciting field. Very little is being done. So this is the only example. We did a workshop in the British Academy a month ago, and there was a lot of like 7 groups on sensory substitution and 1 group on sensory augmentation. But this is a very exciting, and since if you think about it, you take me to a different topics, but it's fun. It's okay. We don't. This is physiology at the advanced part of the course, and we don't really have to fill up the textbook and. So this relates, I think, to a very deep question about the brain and the adaptation to the brain, the plasticity of the brain. Imagine, for example, most of the explanations we have to how the brain organized to receive new types of information is evolutionary examples. So imagine, for example, language. So the fact that we have, for example, dedicated language areas, okay, the Broca and Wernicke, and if we have stroke there, then Broca showed very beautifully that you would lose the ability to speak, to produce the speech. You understand the speech and you can't able to produce it. Okay, so we have dedicated areas to language. So we had enough time during evolution to develop this because we have language for so many years. But imagine, for example, a similar thing, and this is reading script. Stanislas Dian and Laurent Cohen showed very convincingly and beautiful set of experiments in science and some other that you have a brain area in the left visual cortex that responds very, very selectively to a written text of language that you know how to read in. And this is general to any font you know to read in any culture. The consistency of this area is actually pretty amazing. It's more consistent than object-related areas, than the fusiform face area, than all these other areas. It's less than the standard deviation in the peak of these responses to text versus any other object is less than half a centimeter. Okay? But we have text, written text, only 5,000 years. So this is not enough time for evolution. Okay, so what's going on here? So their hypothesis, and it's a beautiful theory, is that there is recycling. So these neurons have a set of characters and connectivity patterns to, for example, language areas and some basic characteristics that is a riddle to try to solve what is this characteristic that made them the best brain areas to adapt the networks to adapt to text when it's appeared. Okay? So I think this is a very cool area, and I will show you also some data on blind reading and using Braille and using sensory substitution. So this is very, very cool field. But to recap to your question, you know, with this exponential growth of technology, and you all heard about singularity, and it's a very sexy field now, etc. Then our brain all the time needs to adapt quicker and quicker to new inputs and new. So I think this would be more and more important. I'm biased, of course. I think you are. Let's stop the recording. You are very biased into other fields, so I need to represent our field also. And there is a new imaging center in Elsc, you know, that we have started, and a magnet to. Okay, I will start to preach. Yeah. Let's 10 minutes. You will give me 10 minutes until the break? Okay, because we are... okay. So what I want to show you now is actually this iMusic algorithm, to train you a little bit about this iMusic algorithm. So the idea is that you have image. The image can contain now also color information. But we'll start with black and white, just like the veteran SSDs. The image would be scanned from left to right, will be delivered, so the sampling rate is much slower than vision. The input would be delivered, would go through some processing unit. The algorithm I will teach you in a second, just the gist of it. And then you hear information and you try to understand. In this case, Max will try to understand the shapes, the colors, the location. He would need to learn to refer it to his body scheme, then produce a motor command and pick up, for, in this example, the red apple. Okay? So I will show you in a second some movies that this is possible. Okay? But first, since it's very not intuitive, we will actually do a training, and there will be an exam. So you need to pay attention. Okay? Now, okay, so let's start with the... Okay, so can you hear it good? Okay, so what you can hear here, so you have a visual image. We start with a very slow, relatively, scan rate. When there is darkness, there is no sound. Each time we hit a pixel that have a sound, we will give a musical note in a shape of a human voice in a choir or choir. Okay, this is how you that sings the line, and when the line stops, they would stop singing. Okay? And if the line is a bit lower, then the sound will be a bit lower. And if the line is still lower, okay, so this is very easy, very simple. Now what happens when we have a vertical line instead of a horizontal line? Now this is a little bit more subtle. So those of you who are more experienced in music, it will sound very dirty. Why it sounds very dirty? Because we have a lot of musical notes coming in the same time. So it's a little bit like pressing all the keyboards of the piano in the same time. Okay? we choose a certain scales and certain distances that any combination would still sound fine, not terrible. So this is part of the optimization we try to do, but it would still sound less pure than a horizontal line. Okay, so let's listen to it. And it will be very brief, because all the pixels come in the same time and then stops. Let's hear it. Oh, just a second. Oh, okay. Before that, if you get the lines going like this, then can you try and sing this? Can you try now and sing? I will give you a cue. Try to think about it, okay? I want you to sing these stairs going down. Okay. 1, 2. I always wanted to be a conductor. 1, 2, 3. Okay, so we have a better choir. Let's listen to it. So very easy to understand, okay? Now the vertical line example. And let's add a horizontal line with a little gap. So you can hear the dirty sound, very brief, gap, and horizontal line. Okay? Excellent. Now let's make the line thicker and without a gap. Can you still hear the more pure horizontal line? Okay, so now, so you hear vertical and pure, short, horizontal line. No questions. Okay. Now you're ready to hear your first word. Do you want to try it without looking and try to read it? Okay, usually I don't do it, but okay, so close your eyes. I will give you a 2-letter word. Just to give you a hint, it will start with this vertical line and horizontal line. Okay? So close your eyes for a second. I would play it for you. Don't open your eyes. I would play it several times. If anyone thinks he got it, he raise his hand with closed eyes. Let's give the others a chance. Okay, so also here there is some variability. I see that most of you can open your eyes and now look at it. You got it? Yeah? Okay. So you are very good. So let's move now to the test. Let's see if you can make the test. Now I would give you a word in English that you never saw. Okay, like you actually you already did it. But let's this is stage 1 in the game. Now let's raise to stage 2. Okay. Now I will give you another word. Okay. And again, it would start with the HI, and then it will have more letters. Okay. Now you don't need to close your eyes because this was already used to do. But actually it's fun. I think we can already do the high example. So let's hear this word. Actually draw it so we see that you are not cheating. Anyone that get it, and raise your hand when you think you got it. So we have 1 already. 2. You can close your eyes if it's something you hear. 1 more. How many letters we have? To those that didn't got it, how many letters? 4, right? So try to listen again, those of you who didn't got it. Without telling me what is the other letters, can you tell me something else about this 4-letter word? Right, the last 2 are the same. So what is the what? Yeah, how many think they got it? Just to check, about half. This is pretty good. Usually it's 1, 2 in the audience. So the word is hill. Tidu, tidu. So you get the low line. Okay, cool. Now what about? So this is nice, but this is only black and white. How can we add color? Anyone have an idea? Let's say we were like brainstorming and grouping and say we want to add color to the image. What? So volume would be different brightness in this case. Okay. So we'll keep it to this, but different volume would be a bit tricky because then you would get You won't be able to hear some colors, and some colors would sound very strong. So this will distort the image. So let's try a different.
This is the instrument. Timbre.
Fantastic, timbre. And this is exactly what we did. I'm not going to tell you exactly the mapping and training about this, but just to get the gist of it, so let's listen, for example, to heel. And now it might sound a little bit weird, the different timbres that we choose and different instruments, but you have a very serious problem here because, for example, I really like saxophone to listen to, and we try to play with saxophone. The problem is that the rise time to get the sound of the saxophone and the decay is very long. And here we want to change between pixels very, very quickly. So we need different timbres that would keep their different characteristics and would be distinguishable between different timbres, even when we switch between the colors very, very quickly. Okay, so this was part of the game here. So still no question. Questions now, because we want to see some images from the brain, the second lesson. Already, I prepared for you 200 slides, and I am in the 10th slide or something like that. This was for a course, or yeah. Anyway, so questions is on the break time on this part. Okay, now what about, so far I only showed you reading, but we want to do it, reading you have Braille. Actually, we're going to talk about Braille and also reading text, just reading the word hill, okay? What if we want to describe something in environment? So for example, we want to draw a hill. Okay, you can get a gist of it, and actually you can feel very quickly if this is a very steep hill or a very, moderate hill. Let's add the little dot. Okay, so what I have next is 4 movies of blind. So what happens in our lab, what we do in this project, and there is a couple of individuals that are involved in this project. I will show you in the pictures after that. They come, and originally they did some training, and then we scan their brain after they managed to do some visual abilities. For example, we test for the classical, I'm sure you heard about the classical organization of the visual cortex to 2 visual streams, the ventral stream and dorsal stream. So we give them exactly the same input, but one time they do shape recognition, and one time they do localization. And we look at the differences and where do they represent shape now in the brain and localization, okay? What we do now is we do it a little bit more organized, and we want to trace, and I show you a lot of data of after they are being trained, but we also have some data which basically trace the changes as they occur. So they would have a scan with hearing this weird sound when it's meaningless, and then they would have it after 2-3 hours of training, and we will have sighted and congenitally blind, and now we also add late blind. And then we will have their brain scanned after 40 hours, and so on and so forth, okay? And then we track how the brain changes, what is the plasticity that is based on the existing connectivity. After 2 hours, you can create new connections. And what is the plasticity that is long-term, and this is long training, and probably creation of new proteins and new synapses and new connections, okay? So this is the plan for the second lesson. We'll start with some movies of blind doing these various things, and then we look on brain imaging of this. If we have time, I would show you a similar project done on cats with deaf cats and the reorganization there with causal relationships. We might show you some TMS on this, and I think we don't manage to get much more than this. So we get to slide number 40. Okay. So let's have a short break and meet here in 12:10. Yeah? Okay.
So questions after the break. What is the reason that you're using sounds with a reverb? Wouldn't it be easier if there wasn't a choir sound but a really limited? You know what I mean? I mean, this sound that we have used now or heard now was a choir, so there are already different kinds of voices in one
Ah,
song.
yeah. Why we use this?
Yeah.
Um, well, it was easier to. We use like really recordings of sound, and it sounds better.
Mhm.
So we wanted the choir because in this case we can have like man and then
Ah,
woman, and then we can stretch more
but
of the
wouldn't one
choir.
single voice be
N-
more useful?
Um,
Or
it might.
okay.
This is what I said. That the exact algorithms and what you use and this is still open to a lot of research and a lot you can think about endless options.
Mm.
And you need to think about ways how to compare them. What is better and what is less. Maybe your suggestion is better,
Mm.
but this is a project in itself.
Okay. Thanks.
טוב, יאללה, נמשיך?
זה כן.
אנחנו אוהבים גם לשתות במעבדה.
כדאי לדבר?
מה? כן, שפר, יש כל מיני היילנד פארק, זה
הכל סינגל מלטי.
הכל סינגל מלטי. לא, לא הכל, יש גם ג'וני ווקר אחד. אבל זה אוף דה מיקרופון. טוב, אז בואו נראה קצת מה, אז העברים באים לתוכנית של כ-70 שעות. Yes, sorry. So you didn't miss anything. So we are all back, or you want to call them? We are 2 minutes. אני רואה שאפילו לא הספקתם לאכול. Okay, so they would come to the lab, and there is a training program that we all the time try to improve. Now we try to make it as a self-training, because at the moment it's only they can only come to the lab and being trained, and they come for once a week or once every 2 weeks for 1 hour or 2, get training, and let's see. So after 1 hour, they can start recognizing very, very simple shapes and colors, and localize them. 1-2 hours of training. And they start without the camera. They start just to receive the input, and it's localized. After that, they need to start learning, and all the data that I'm going to show you in this hour are from congenitally blind individuals. Okay, so we talk about people that were checked by neuro-ophthalmologists, and it's very clear from their medical records and from testing that they didn't have any visual experience, so they are blind from birth, and they are fully blind. They don't have any light perception. A lot of people that are considered legally blind do have some form of light. So if you close or open the light, they might say something, but they don't have any form of localization or of shape information. Okay, so this is all the individuals that I'm going to present here are fully congenitally blind. Okay. After 10 to 30 hours of training, they can already, so you see here Chamutal, and she's congenitally blind. She has this camera here, and she looks in the image to get the information. She understands when is the red. She really likes red apples, so she grabs for the red apple and pick it up. In this case, she needs to localize where is the bottle of Sprite. Okay, so you see, it's slower than vision, so it takes several seconds, 3, 4, 5 seconds. She recognizes, and then she combines it with her body. You see, when it's on the left, she would reach for the left hand, and when it's for the right, she reach now with the right hand. So it's really, this is the second topic that I wanted to talk about with you, mapping of the body and the motor system, the somatosensory system, etc. But you see that she connects her body scheme with the visual information outside there. Okay, so this is pretty cool. And then they can, so you see here reading, but also as the training progress, we try to move from practice in front of the computer to practice in a natural environment. Okay, so this is this case, and it's not by accident. The floor is very white, the shoes is very dark, so there is very good contrast. And she tries to recognize it and reach for it. In this case, this is training of different categories that we know in advance they have some specialization in the brain, like faces and the fusiform face area, body postures for the exercise body areas, movement, reading, so on and so forth. Okay. And I think the coolest example and the movie I like the most to show is in the case of the face category. I think what is nice in this movie is that you see, first of all, the interaction between the trainer, in this case Lior, Reich in the lab, and Elsc Amit that led in beginning the training, and then the neuroimaging, and now there is other people involved. So you can see the interaction with the trainers, and you also see how the training builds up from very simple feature of the face in line drawing to more and more complex to real faces in a natural environment. Okay? So this will be the last movie, and then we'll talk a little bit about the brain.
זה אף.
אוקיי, מאוד.
שומעים את שני הנחירה, את שני
איך אתה
הנחיריים.
שומע את הנחיריים?
כמו קליקים, טוב, לא. מה זה גם? וואי, נראה לי שומעים את העיניים ואת האף.
יפה.
So you see in the beginning
ואתה
there is
שומע
no
אולי עוד
camera.
משהו?
Because later they need to learn to fixate, for example. It's
שנייה.
very difficult to learn to fixate.
נראה לי שגם הגבות מופיעות.
מעולה, כל הכבוד. בוא תנסה עכשיו להגיד לי.
העצוב זה השני.
יפה מאוד.
If you think about it, social cues, it's something critical that they miss. So first of all, they don't have a grasp of far people and where they're located and the body postures, but also social cues. So in the second, here you see the full setup, you see the camera, you see the headphones. So some facial expressions she knows, for example, smile is innate, but some she really needs to learn the shape. So she learns it by touch. And now she would look at Ella. She has the reconstruction, in this case of the voice algorithm. This is the black and white algorithm better developed by people here. And she would get the input of that comes from far. Visceral active vision, you remember we talked about it? So she knows this is Ella outside and that she's inside. Of course, this is what Hagai Berman likes to call the best typical example.
לפי מה? ועכשיו?
No, it's of course as you get farther away,
מעולה.
smaller,
מעולה.
but you can do things that are more far away. Okay, so now to the interesting question, what happened in the visual cortex without visual experience, okay, and the brain in general when we learn this information and without learning it. So first of all, I want to show you this image because it's very important. I'm going to show you later, and this is what you usually do in brain imaging. You are not looking at, you're giving pictures and you look at the brain in general. You need to choose and do a very controlled experiment because you want to say something, let's say, about the processing of reading. So you want to differentiate activation to reading. If you just give them, for example, the word heal, and you look at it in vision, then you have, let's say, the visual word form, the area that is very selective to vision, but you also have V1, V2, and you have all the visual input. So you don't know if the processing is related to the reading or to the visual input or to the line or to the line junction. So you need various controls. So you would need to contrast it. So a lot of what I will show you later is contrast. For example, we find the human homologue to area IT, which is the ventral stream, the shape processing, by taking real objects and scrambling them, okay, and contrasting them to scramble or contrasting them to textures, etc., and then try to find something that is more selective to objects, okay? So you would see later contrast. This is what I'm showing you here is the brain responses to the heel, but it can be the healed road, it can be a real object, it can be space. It's a very similar picture. And now I'm showing it to you versus baseline, okay? Why is it important? Because what you see when you look at the brain, so you scan the brain while they're doing this, like what you did reading the heel or an object, you see massive activation in auditory cortex, and this makes sense. So you get activation in A1 and all the areas. But you also get activation in all these transition areas, in almost in the entire visual cortex. I'm going to show you now more elaborated and detailed objects, images of this. So it seems like they recruit both their entire or a lot of their auditory system and their visual system. Okay? Now, the second piece of information that there is, and this is usually something that you don't realize you can do with brain imaging, and of course it's not optimal. Brain imaging is very good to get high-resolution images of the human brain while people are doing various things, so this is something that's very difficult to do with animal research. But of course, you have to pay in the form of very poor time resolution, okay? So in principle, you get an image of contrast. They will do various things like object recognition. They will do or reading. They will do various things like scrambling the words or scrambling the images. And you contrast a long period of object recognition with long periods of things without, and you will look for the large-scale organization of object recognition. Or you can map very accurately by using retinotopic mapping or moving from the center to the periphery, all the visual areas, V1, V2, all the homologs to the primates, very, very accurately. In 10 minutes, you can finish to localize all the visual areas and then start to ask questions about them in the human brain. So, for example, what happens when we imagine a line versus when we see a line, okay? So this also, you can ask things that very difficult to ask in animals, you pay for the time. But what is cool is that in some cases, and for some reason people don't use it too often, and we try to start to use it more and more in the lab, in some cases, when the processing that you are doing in the brain is slow enough, so usually, for example, when I would flash you an object, very, very quickly you can recognize it, and in 500 milliseconds everything is finished, okay? Even the recognition and all the rest. So if you sample one every and usually you sampled one every 3 seconds, okay? Now you can sample you can sample the entire brain in once per second, or you can zoom in some areas and even higher resolution. But let's say now I'm doing it with sensory substitution, and I'm looking at I'm getting the objects by the sound, and it takes me 4 or 5 seconds to process and recognize that this is the cup, this is the face, this is the word, etc. So now we can actually look at the chronometry of this process, okay? And what is cool is that the way you used to do retinotopic mapping, or mapping the body, in this case it's a work by Noah Schaya, which is a student in the ICNC, and Uri Hertz, they're both. So they used the notion that you can do something that is relatively slow. So either the process is slow, like in the case of object recognition that I'll show you in a second, or you do the experiment slow enough so the resolution of the MRI is good enough to capture it. So what Noah did in this experiment, she asked the subject to move for several seconds each body part, 20 different body parts, or in different experiments we touch the body part, or we ask them to imagine a touch in the body parts. But the touch is being done in a very organized sequence. So in one experiment, it would start with the leg and would continue. It will take 20 seconds to reach all the way in a gradual manner to the face and to the mouth, okay? In vision, you can start with a foveal stimulation and go to the more periphery, and you will get these maps going from the posterior part to the anterior part, mapping all the way from the center to periphery. You repeat this many times, always in exactly the same order. Of course, you will need to do the reverse experiment. You would start with the face and reach to the legs, or you start in the middle of the body, and so it needs to be cyclic. And then you would use either or both, that's the best, Fourier analysis, okay? So what would you get if you get Fourier analysis? You would look on each voxel independently, let's say 1 by 1 by 1 or 3 by 3 by 3, and you ask which voxels across the entire brain have a lot of power, a lot of energy in the frequency that you stimulated, 1 divided by the cycle that you have, okay? So then you get all the areas that respond to this cycle, okay? Of course, we can have slower wave, we can have noise in high frequency, but the things that respond in this frequency, they have some relation. Let's say the leg area in the Penfield homunculus would all the time respond here, right? So it will have a lot of energy in this frequency. But what is cool is that you can then take this in all these voxels and now color each voxel according to the phase. Okay? So early phases will get the blue color, so it corresponds to the leg. And later colors will respond to the mouth, and they will get a red color. So you get a beautiful reconstruction of the Penfield homunculus. And what she found really cool, we are not going to talk about it, is that you get also a negative bald homunculus. So you get, when you move the mouse, you get deactivation that is cyclic to the legs at the same time that you are doing mouse movement. And you can actually predict which body, if I will give you now a random movement, she can predict both based on the positive and the negative signals if it's moved. And it actually says that the entire homunculus, Penfield homunculus, is active when we are doing a movement. But some of the neurons showing increased activation and some of the neurons showing increased significant deactivation, okay? The second very cool thing she found is that there is actually a family of homunculi, about 10 very accurate homunculi. This is still not published. So the first she published in PNAS, and there is a additional work she is now doing. And there's actually a family of homunculi that you can, in high resolution, map in the human brain and start asking what, in vision, we know that there is transformation. And there is decades of research about the transformation that are being done from each area and what it's representing, so we can start test it in other systems. The somatosensory system, for mental imagery, for the motor system, for the visual system, and interactions between them, okay? Now let's go back to the example of the sensory substitution. So let's say I'm giving you this cap and it takes you 4 or 5 seconds to do it, and I give it to you 20 times, okay? And each time it takes you 4 or 5 seconds, or we can give you different objects. Now we can go back, ask which areas are responding to this frequency, and then we can again look in which areas respond first, and then where the information flows in the brain. This is something you usually think about EEG. You wouldn't think about high-resolution MRI. But if the processing is slow enough and your sampling rate is higher than this, then you can start looking at the chronometry. And this is the next movie I'm going to show you. So this is what I described. You can also use cross-correlation. So you can have a model of the typical hemodynamic response. So usually, I won't get into the physics and the details of it. You would have neurons active very strong. You will have oxygen consumption. And then the brain somehow knows very accurately to overcompensate, to send extra oxygen. And this is the peak in the signal that you get. And then you get a little drop in the oxygen after it's being washed away. And then you go back to baseline. So we can have this model and now shift it and look for cross-correlation. Shift it in time and then map the activation as they progress in the brain, the information flow by this model. Okay? So let's look at and you get this beautiful Penfield map. So you remember the homunculi, you put your legs here, and you lay down like this. Yes?
Can you just define the Penfield map quickly?
Yeah, so the Penfield, this is supposed to know already from Physiology A, but the Penfield map is basically, imagine that there is a person, so let's talk about the neurons, and in this case, the imaging that reflects the neurons, okay? So you have here neurons that when you move your legs, they are very active, but they are not active, or they are actually deactivated, what Noah showed, when you move your mouth, your lips. Now I'm talking, okay? Now if you move a little bit laterally, you will have representation of the leg. And you move a little bit, you have the representation of the thighs. Then you have the representation of the arms, of each finger. Then of the mouth, the lips, and then so on and so forth. So you have actually an internal map that represents the envisioned external world, the visual fields. In the case of the somatosensory, the motor system, the body in relation to movement, and in the case of the somatosensory system, in touch. What is very interesting about this homunculus is that it's not proportional to the actual physical world, but to what is important in terms of task, or in terms of what we need to achieve. So, for example, there is a huge part that is dedicated to this finger, more than the entire back. Okay? Or the area to the tongue is more than the entire area of the body from here to here. So it's unproportional, and there is more neurons dedicated to this. So this is, to do this, you need a lot of, many years in animal research. Now that we know them, but we can look now for new maps and learn this transformation, you can do in 8 minutes of imaging in the brain. And then you can also try to ask where is the homunculus in the subthalamic nucleus if you have high enough field. So there is some magnets in 7 Tesla. So now there is research to try to see if we can map the homunculi there. So you can target deep brain stimulation in terms of Parkinson and be much more accurate. It would shorten the surgery in hours. So this is critical. This is just to give you a taste of the 100 slides we are not going to show. But as I said, we can have you can see here a phase movie of the movements of the body, in this case the motor, and then how the information flows, the phase flows. So this is a representation of the phase, how the information flows as we move different body parts. Okay? This is a bit in slow motion. I think the computer suffered from putting so many slides. But this is so this is what happened in the brain when the congenitally blind learn to do reading or object recognition using auditory sensory substitution. So you see that first the information goes to A1 and the areas around it. And after a lot of research that I'm not going to describe you today, I won't have time, and some of it is unpublished, what we think is happening is that the A1 and the areas around it are doing quick Fourier transform, understanding what is the frequencies and what is the power, but then it sends the information to the visual system where it's building the image and doing the actual spatial combinations to do object recognition or to do various things. Why do we think that this is what happened? I will show you some.
Why does the brain do it?
Well, that we are
It says
right. So
why does the brain?
right. So when we started initially, you have various hypotheses. One is that it will use the auditory system. So you have areas dedicated to reconstructing the envelope and the shapes and the locations in the world, etc. The other hypothesis is that it will move the information to multimodal, high-order areas in the brain that converge usually sound, audition, vision, and maybe also touch, like in the case of the cat that I showed you, the work of Joseph Rauchecker in the blind cat. And what we found, and let me show you some of the evidence for this in a little bit more convincing manner, is that most of what is happening is that the information is being sent to the visual system, and there the visual system is doing the actual task. Okay? That's what happened. We are experimentalists, so we do the experiments, and let's see the evidence. So first of all, this movie shows you that this is what's happening in terms of general activation without good controls. Now let's start doing the experiments in a little bit more organized fashion and with all the controls. Before I'm going to show you all the details, we are going to, I'm not sure how much we are going to get into the details and all the controls, etc. Let's summarize two things that we have a lot of experience in both in the visual system and in sensory substitution. So we know, for example, from the work of Rafi Malach and hundreds of replications in various labs in the world, that when you do object recognition versus scrambling of objects or textures, or etc., you get a very, very consistent activation in the occipitotemporal cortex, which actually fits very nicely with the ventral stream in the primates and in the cats and in non-primates of the ventral stream of IT cortex, for example. So it's a very preserved location in the ventral stream, okay? And it's very consistent. You see any object, it can be abstract, it can be something you saw before or not. As long as you decipher the shape, 2D or 3D, you get this bilateral activation, okay? Now, when you look at congenitally blind, and in this case, we also did training of sighted and of late blind. After 40 hours of training, you get the specialization. So we replicated exactly the same experiment that we did in the sighted with object recognition versus textures, transformed them into sounds, tested the behavior that they can manage to do it perfectly after this 40 hours of training, give them the stimuli, and the activation goes into the lateral occipital cortex. So if I would show you objects and the textures versus rest baseline, of course you get massive activation in A1 and around it. It's not that they don't hear this. They don't use their auditory system. But when you contrast objects with non-objects, with all the controls, you get this activation. So this is a good question. At that moment, we didn't do the very long experience of imaging them in various points, but if we manage, I will show you some data. Now we have collected all this data on several experiments, and we finished to analyze one experiment of desegregation for the ventral and dorsal processing. I will show you some of this data, hopefully. Okay? I'm just doing an overview because I don't know how much I would get, and this is published, so you can download. You can go to our website if you are interested, of course. You can download the papers and read all the details. There is all the description and all the controls, etc., etc. Now, what is cool is that recently we also repeated this experiment by touch, okay? And we again get recruitment and very selective activation in the same area, in the congenitally blind, but also in sighted. Okay. Now, let's go to look if there is functional specialization. We start to look into this networks of these areas that, in addition to this general object area that is responsive to any object, you have also some areas that show some selectivity, and there is a lot of debate how much it's selective, etc., and how much is this area. There is no debate about the data itself, but about the meaning of it, etc., is, for example, this visual workfront area that I showed you. So what you see here is results from experiments that Elsc and Amit, with a couple of some others in the lab, ran, in which the blind read text, but they also do recognition, they do categorization for several others. So they look at objects, general objects, they look at faces, they look at houses, and they manage to categorize, to say this is a face, this is the letters, this is etc. What you see here is a contrast, and we had a group of sighted that run exactly the same experiment, but by vision. So what you see here is a very clean group result, and there is a lot of statistics involved, random effects, very carefully controlled for multiple comparisons. So you get a very selective activation for the written text in a language that we know how to read versus all the other categories. Okay? So faces, houses. So this is a very stringent control, and you get indeed a very selective activation in the left ventral visual cortex. Okay? Now, what is really cool is that when you repeat these experiments in the blind after they learn to do what you did, read the heel and recognize also these various categories, you get exactly the same result. So you get specialization for this text, for this combining. What do we do when we read? We take certain symbols, we assign to them a certain phonology, and we build from them the basic units of reading. Okay? And if you think about it, this is exactly what they are learning to do. Now, just to make sure that this is, not to make sure, actually, this was a previous study that we did. We started with something which is a bit more easy. You recruit congenitally blind individuals that are proficient Braille readers. Okay? You let them read Braille versus symbols that don't have any meaning. Okay? So also in touch, this is the control in this case. And some other controls. You let them hear the words, you let them do various things. And you do this contrast, and this is a study by Leo Reich from the lab. You can also download this with Braille reading, and you get exactly the same results. Okay? Not only you get these results in the case of the Braille reading, and also in the other case, but I won't have time to show you, is that if you look at the peak across the entire brain in sighted reading by vision or blind reading by Braille, and you plot all them on a normalized brain, so you normalize for different sizes. Okay, Thaler proposed this normalization. Then both you in the vision eye, if we remove the labels, or K-means algorithm, for example, that try to separate it to two groups, can differentiate if this is blind reading by Braille or sighted reading by vision. Okay? So it looks like quite a consistent result. So let's so I gave you already the bottom line of the experiment. Let's look. We have some time. I'm going to run very quickly on this. Okay? So if there is really something you don't understand, we will. So we used in this case this set of experiments. I'm going to show you the grayscale algorithm that is called the Voice, that is developed by Peter Mayer. So it's not the. We are now running a set of experiments with the color algorithm, that is the iMusic that is developed in lab, and we ask, for example, what happened when blind learn to differentiate different colors when they never saw colors? So after they do the training on the timber, they can distinguish different colors, okay? So what happened in their brain? This is a project by Sami Aboud, but it's not fully analyzed, and I'm not going to show you. But first of all, we tested their visual acuity. We ran a vision test on them using the sound. This is Elad did, just to see how accurate they are or what is their visual acuity. And you can see that 75% of the blind managed to reach visual acuity, which is comparable to the threshold that defines someone that is passing from being considered legally blind to severe visually impaired. So it's 20 by 400. You divide by 6, you get how it's used here, but the WHO used this. And this corresponds to about 4,000 pixels. So in principle, they can, in 40 hours, reach a level which they can have 4,000 pixels. But of course, that doesn't mean they can really combine these pixels into meaningful objects. So we, of course, run also this behavior. And you can see here, they can reach much more significant, about 80% success in categorizing these various objects. Okay? And now, having this being achieved, we can look at the brain activation. So now I'm showing you a little bit more. At least one experiment we will try to look at the data in an organized manner. Okay, not just summary. So let's use these experiments to show you some of the data analysis and the contrast and how we look in the brain with them. So what you see here is the activation for letters, for reading letters, in sighted using vision. Okay? So here we don't contrast it to any other object, and we used very strict, corrected, and et cetera, and you see very significant activation in the ventral visual stream. Now, of course, if you reduce the threshold, you would start having the activation goes backward. But the areas, the neurons that fire the strongest, and you will see that actually they are activated in both sides. I showed you a very, very clean activation to letters only in the left hemisphere. But when you do this versus baseline, you get this bilateral activation. You get other areas in the prefrontal cortex, parietal cortex, etc. But also you get quite a clean picture. There is nothing in the auditory system. There is nothing in other areas, dorsal stream, etc., etc. Now let's look at the same when blind are reading the heel, like you did, okay, versus baseline. So what you get is a very strong activation. This is the Hessel gyrus, okay? This is the human homologue in first approximation to A1. So we have very strong activation in A1 and all the areas of the pleurale around it. But you see that not less, you get some gap actually here, and you get huge response exactly in the same area. So very similar areas. You can see the cross here is the, after averaging a lot of sighted individuals, Stanislas Diane and Laurent Cohen came with the average location of the visual word form area. And you can see that in both cases there is a little bit of a shift, but in both cases they fall, the activation falls very, very, like around the visual word form area. Okay? But also you can see here that it's bilateral. And again, I just want to remind you, we talk about people that didn't have any visual experience in their life. Okay? Now we look for the selectivity. We want voxels that are significantly more active to letters versus faces, body parts, all the categories that I showed you before. And now you get a much more selective activation in the case of the sighted, and lot less, and what is actually quite surprised us, that it's even more clean than the results of the cited. This is, okay, this is not the main point, but it's actually very, very clean in the case of reading. So maybe, well, I won't elaborate too much because it's speculative, but what is very clear is that we get a peak around the visual wolfram area in the left hemisphere, regardless of visual experience. This is true also if you look at probabilistic mapping. So now you can repeat this trick in all the individual subjects. Usually in MRI, you won't see people showing you single subject analysis because it's it's relatively noisy. Of course, you need to average. Like in single units, you need to average hundreds of trials to get, and so on and so forth. But actually, what is cool, it's so consistent that if you normalize the brain according to Thaler-Reich, and you repeat this analysis on a single subject level, and you ask which voxels are overlapping in 100% of the subjects, you get this only in the visual wolfram area, both in the sighted and in the blind. And what was even more surprising is if you do the same trick, but now in the group level, and you contrast letters to textures, to faces, to houses, to objects, to body parts, you do this statistical test in the 0.05 corrected for multiple comparison, and now you do a conjunction analysis. So you ask to present only voxels that passed all 5 statistical tests. Okay, so this is the conjunction analysis, and you get 100% overlap in the visual wolfram area. So this is actually the strongest result I ever got in neuroimaging. Okay, so I will finish this this experiment and just summarize. Okay, and I will just mention one work of the 5 minutes, and we will finish. Okay. But this is, I think, a really cool example. This is only a case study, and all this work is already published, so you can download and take a look at it. We had one of our subjects, so all the subjects that I showed you are congenitally blind individuals, and all of them, and we tested them in adulthood, and all of them are braille readers. But all of them also know to recognize the Hebrew alphabet with their shapes. Some of them do it because there is embossed letter, some to communicate. For example, Chantal have a child, and is a sighted child, so she learns, etc., etc. So we know that they learn Braille, and after a lot of training in other scripts, or in the sensory substitution, and after that in the sensory substitution, they develop this selectivity. But what happens if you teach a new script, a new shapes, and you don't give enough time for new connection to form? So what we did in this experiment, so this was pretty cool because one of our subjects knew to read Braille, okay, but she didn't know the shapes of the alphabet. So what we did with her, we did the entire training the same as all the other subjects, but we left out the training of the reading category. Okay? So this is clear? So we did all the training of the object, of all the rest, lines, etc., but no Hebrew alphabet. Now, what we did was to scan her brain on a Braille letters, on these symbols that are meaningless in Braille, as a control for the touch and for the dots, etc. And we gave them 2 types of letters: 5 letters, a group of 5 letters, and a group of 5 other letters. Now, what is the difference between these 2 groups? In the first experiment that she did in the scanner, both of them were meaningless to her. Okay? Then we took her out of the scanner, and for 2 hours, she already knew to use the sensory substitution very well, so it was easy to teach her. For 2 hours, we trained her to recognize 5 letters. Let's say one of them is the W. And 5 letters, we didn't train her. So these are still, she understands the shape, but they are meaningless in terms of their phonology, and they are meaningless for reading. Okay? And we repeated exactly the same experiment after these 2 hours. So this is more similar to the example I gave you with the FedEx. You have like this some types of learning and plasticity that is very quick and stays there. So this is to tap into this type of plasticity. I won't go, we do a little detour. This is the results of the Braille reading versus the control, just this part. So this is the results of the peaks, by the way. This is the blind is the red, sighted is the green, and this is without the tags. So you see the consistency. This is the other study I mentioned. Let's keep this other work on language and memory and go back to the results. By the way, if you give sighted these sounds and you teach them to do the algorithm, they recruit the LOC, all of them, the individual subjects. If you take a different group and you let them learn just association, so you give them, this is an arbitrary experiment, you let them have this weird sound of a horse, let's say, and you tell them this weird sound, you need to learn arbitrary association. You don't tell them anything about sensory substitution. You tell them this sound represents a horse. This sound represents hammer, etc. And we repeat exactly the same experiment. So the physical simulation is exactly the same, but they don't learn to decipher shape. They get the sounds. And you do this subtraction between objects and scramble. In none of them you get LOC. You get higher order multisensory areas. Okay. So, and actually, I have to show you this movie. This is also anecdotal, but this is what is cool is that it shows causal evidence. So far, I didn't show you too much causal. It's all correlational. Okay, so you can show correlation to behavior. You can show. So what we did in this experiment is one of the subjects that we had, and this is was done in the lab of Alvaro Pascual Leone with Lot Fimerva. So we sent them the data imaging that we had on these subjects of the activation in the visual cortex, and they did something that is called TMS. Did you ever saw TMS? You saw okay, for those of you interested in the data, did you ever saw speech arrest in TMS? Okay, so those of you who are interested, I would show it to you in the end of these 5 minutes. But this is really cool. You can make somebody that tries to speak, you do repetitive TMS, and you can make him stop being able to produce the word. Although he's, and the idea is that you inject noise into the area and the connected area to this, the connected neuron to this area that process this information, and you look at the behavioral correlates. Now, usually you don't get any effect with TMS. It's very subtle. You need to do very controlled experiment in threshold to get some little influence. It's very difficult to get good results with TMS. But when it's, in the few cases it's work, like in the stimulating the motor homunculi, you can twitch the hand or the leg or the different muscles. So this is very convincing. Speech arrest is another example. And this example is using TMS in a different mode. And this is the mode you can do it in the motor system, for example. You use a certain frequency, and you move the hand with a different some amplitude. Then you do 10 minutes of repetitive stimulation in the same area. And now you give exactly the same stimulation. The movement is much weaker. So you reduce you suppress this area. And after 10 minutes, this is washed away, and you get back the, okay? So you can actually manipulate the cortical excitability. Today, there is new other methods, for example, TDCS, which uses direct stimulation. It's much more subtle, but you can increase the excitability or decrease in a given brain area using this in humans. So what was done in this experiment is that first you would see a pet using this camera. She's looking at the object. She's detecting.
Object is detected.
This is very difficult to trust, yeah? 1, 2, 3, 4, 5, 6.
For you.
So now you can, understand
No
what I
connector.
showed you the movie in
The
6
object is now in front of
seconds.
you.
Now she has 10 minutes of
find that
TMS,
edge.
and she repeats the same thing.
There's the edge.
And I can go on and go on and go on, and she can't recognize. You do washout, she can recognize it. We did it for 5 various objects. So this is really shows that not only they activate the object areas, but it's functionally relevant. This is very anecdotal. I would skip all these things. I would just mention very briefly that in one of the experiments, we gave exactly the same input, and then we asked them in one case to do shape analysis and in one location analysis. So one to ignore in one case we asked them to report if inside the scanner if the shapes are curved or with angles, and in the other we asked them to say and if it's the same or different. And in the second we asked them to say something about the location. It was the same location or different location. Okay? And what we found is that not only that we got recruitment of the IT cortex for shape versus location, this was the peak across the entire brain, and in the dorsal stream for the location versus shape, but if you now look on which areas are being enhanced during training, you would see, and this is still unpublished result, you can see that if you do shape and you contrast the long training with the brief training, so the neurons that are responding more and more to shape are the ventral stream neurons that are going back into the retinotopic areas. And it's the same in the dorsal stream, and also recruited cerebellum, but this is a little bit weaker. And this is very important because, and this is the segregation too, because in sight recovery, if you use these other approaches, it's very difficult to get shape performance. So we're actually thinking, and I would maybe, so, okay, let's not talk about sight restoration. It's enough in one lesson. So let me just summarize. And this is now the more controversial part. Okay, this is our theory. There is some people that agree with it, some not, and there is some replication in different labs, and some others suggesting other theories close to it. But basically, what we can summarize is that visual areas, including the visual word from area, which is selective for reading and for letter strings, the extra stride body area for body parts, the LOC for object cognition, and the selectivity to the two streams. And also in other groups, they showed very nicely that the MT for movement in the dorsal stream, results by Ravsheker, Collignon, De Volder, and Yer group, appears to be related to the task that you are doing. So if it's LOC, it's deciphering the shape of things, regardless of all this. And this is quite, very different from what you would find in the textbook. So regardless, first of all, of sensory modality, so it doesn't matter if this is done by vision, by touch, or by audition. If you do this task of shape deciphering, you will get it. If you do the task of reading, you would get the visual word from area. What is even more bizarre and crazy is that it's regardless of visual experience. So you don't, even if you don't let the system develop according to the visual input, you would still get segregation to two visual streams. You would still get segregation for object cognition in the IT cortex. You would still get segregation for the reading network. In some cases, and this is still anecdotal, it's regardless of the age of learning a new modality. So you don't need to learn in the period that your brain is very plastic, like in the classical critical period. In our case, we can have somebody that is 35, 40, that learned this new script. You would still get this selectivity to the reading. This is still very anecdotal, so it's just this one case that we reported in there. And long-term training on the script or on objects. So we show that in some cases, even after 2-3 hours of training with these new devices, you start to get these selectivities. So there is something that is innate. So how can we explain this? So what we suggest is that the classical view of looking at the brain as a sensory machine is not accurate enough. So we suggest the brain is a flexible sensory modality independent task machine. Okay, and the thing is that you would ask then why in all these thousands of experiments it appears that this area responds to vision. You do these experiments in the cat, you get visual responses. You do it in the humans, and this is thousands and thousands of papers. So what we try to argue is that it's only an illusion that these are visual areas. Why they look as if they are visual areas? Because usually, if you do shape analysis, the best modality to do it is vision. It gives you the best Bayesian predictor on what is there in the environment. So it looks like it's a visual area. But if you prevent the visual area and you do it by touch inside it, you will get the same selectivity. If you do it even without visual experience, you do it with congenitally blind by touch or by braille or by the sensory substitution, you still get the same selectivity. So it's illusion that the ventral stream is vision. It's not vision. It's vision in the case that vision provides you the information to read. It gives you the information to, but if you achieve the same task using a different modality, the selectivity is in this sense. Okay? Okay, I won't have time to show you, but this is amazing results that generalize this in animal research. This exactly the same view. Recent work by Steve Lomber lab, and what he's doing is pretty amazing. I won't have time to show you, so I just jump to the end. You can very briefly look at it. So it's a detection of movement and detection of localization using vision in deaf cats. And what is really cool is that he cool certain auditory areas and show that it have better movement detection only in the periphery, in the visual periphery, in a certain auditory area that is selective for auditory motion in the hearing cat. And it shows double dissociation for localization in a different area. You see here the cooling maps. This is the advantage of animal studies. It's so clean. It's so, and you can actually see exactly the cortical area. So I'm not saying anything against animal research like they might like to say about human studies. But I think you can do a lot of super cool things using the human brain and advanced imaging techniques. And you can see here the cooling in the two different auditory areas, and here is the causal relationship